Hello there. You want to learn about protein synthesis? Well, I have a few friends that can help us out. Let's go meet our scientists. Here are our scientists, 1 and 2. But wait, where's 2?
I messed up. That was my fault. 3, 2, 1.
Do a little refresher on DNA structure. Scientists, here we see a classic strand of the DNA structure. This is also called the double helix. Next, we see the double helix unwound. The double helix unwound. Correct, scientist number 2. Finally, if we break the DNA strand down to its individual rungs, very good, scientist number 2. The rungs on the ladder are made up of 2 different nucleotide bound together by hydrogen bonds. The base pairs are adenine and thymine, cytosine and guanine. Do a little refresher on DNA structure. Now let's go over the process of transcription. There are 4 steps of transcription. The first step is that RNA polymerase binds to promoter DNA, which begins the process of transcription. Next, RNA polymerase creates a helicase bubble and breaks the hydrogen bond between these base pairs, typically unzipping the DNA pair. Step 3, RNA polymerase adds complementary base pairs to the leftover RNA. This is good for the 3' 5', which is the leading strand, but for the 5' 3', they have to go in with Okazaki fragments, which makes the process slower so that they can eventually recreate a new strand. Thus, the DNA is preserved and the leftover strand of RNA is taken to the endoplasmic reticulum. Now let's talk about the next step in protein synthesis, translation. Step 1. mRNA enters the endoplasmic reticulum and the ribosome is attached. Step 2. A tRNA carrying a start codon enters the ribosome. Step 3. The amino acid commences the polypeptide. Step 4. The ribosome travels down the mRNA as more tRNA arrive with matching codons. Amino acids grouped together, continuing polypeptide. Step 5. Process continues until a stop codon arrives. The polypeptide is released and the ribosome detaches.
Two, 1.
I've been to the nucleus. It's really cool. Transcription is really neat.
After that, you should check out the endoplasmic reticulum. The process of translation is really neat.
Now that you know about transcription and translation, let's talk a little bit about sickle cell.
2, 1.
One downside of sickle cell is that because of the crescent moon shape, your blood cells can clog your capillaries, cutting off your circulation. An upside, however, is that sickle cell can help prevent you from malaria.
2, 1, go.
In a regular DNA strand, the adenine and thymine are in their natural places. However, in the sickle cell mutated gene, the thymine and adenine are switched. That causes the RNA to be affected. The adenine then changes to the uracil. That makes the entire protein mutated. Now let's move on to beta globin. Scientists! Beta globin is a component of a larger protein called hemoglobin, which is located inside red blood cells in adults. Hemoglobin normally consists of 4 protein subunits. true subunits of beta globin and true subunits of another protein called alpha globin. Hemoglobin within red blood cells binds to oxygen molecules in the lungs. These cells then travel through the bloodstream and deliver oxygen to tissues throughout the body. People who have diabetes need to get their hemoglobin tested with HbA1 that tells you their average level of blood sugar for the past 2 to 3 months. And that is all you need to know about protein synthesis.